So I will talk in the next 45 minutes or so a little bit on the basics of the genetics of complex disorders, complex neurologic disorders, and Parkinson's disease, as you will see, is a complex neurologic disorder. It does not have a single cause. It's rather a whole web of causes. I will discuss the genetic architecture of Parkinson's disease. We'll show you some of the monogenic forms of Parkinson's disease, but also discuss the genetic risk factors that contribute to the sporadic form of the disease. Also allude to genetic testing and to the emerging mutation-directed treatments, which are currently in development. There are clinical trials targeted at the defective genes in the genetic forms, which might also be of relevance in the sporadic disease, and we'll try to get a hold on all of these complex issues. So the learning objectives will be that you should be, at the end of the webinar, be able to recognize patients with a high probability for a genetic form of Parkinson's disease. There's no way that you can really predict, and I can't either anyway, but there are some specific features that point to the involvement of specific genes and pathways. You will also hopefully be able to describe the major pathogenic pathways that are involved, molecular pathways in the genetic forms, and also their role in sporadic disease. Explain the concept of a polygenic risk score in the sporadic disease, and discuss the current state of targeted therapeutic trials in Parkinson's disease based on these genetic findings. And as you know, you will be able to ask questions in the chat, and we'll discuss that at the end. So let's start with the genetic basis. It has probably been quite a while for most of you that your genetics courses at medical school or wherever you got your education are over. So just to refresh you on that. The human genome consists of about 3 billion base pairs. The actual genetic information that is coding for genes there is organized in 22,000, roughly 22,000 genes, which themselves are composed of a total of 180,000 exons. So that is the coding sequence of the human genome, and that is only about 1 to 2% of all the 3 million base pairs. So only about 50 million base pairs are actually coding for proteins. So all the 98% of the remaining genome is not coding for proteins, but it has a very important function, namely in the regulation of the expression of the coding genes. Probably not all of those non-coding segments of the genome, but an increasing proportion of them are really involved in the regulation. And that's very important to know, because a complex creature like a human being is driven, the development is driven by those 22,000 genes, and can be driven only because those genes are very, very highly regulated. Now, if we look at the genome of an individual, and that's just an arbitrary example, let's say those are 5 different little segments from the genome. You can see the alphabet, the genetic alphabet, the nucleotides, G, A, G, A, C, C, A, A, and so on. And you can see that in many parts of the genome, they are identical from person to person and from chromosome to chromosome. And in fact, about 99.9% of all the bases of those 3 billion base pairs are identical from one individual to the next. So we're very, very, very much alike from person to person. However, there are some differences, and that's, of course, those differences are very important. And those differences in the reference genome are called variants, and they are not bad or good, they're just variants in the first instance. And some of those variants are relatively common, like here in this position. You see that roughly half of the individuals or in half of the chromosomes, there is an A, and on the other half, there is a G. So you can have a GG, an AG heterozygous, or an AA, all in the same position. In some other positions, there are very rare variants. Only one individual here has an A in this position. All the others have a G. So there are rare variants and there are common variants. The rare variants can be very rare, only 1 in 1,000, 1 in 10,000, 1 in 100,000. The common can be very common and they can go up to about 50%. So common variants and rare variants. That's a very important concept. On average, about 1 of 500 to 1,000 bases differ. So 99.9% are identical, 0.1% differ, and those different variants can have variable frequencies. So a total human genome carries about 3 billion variants, 3 million variants. There's an interesting relationship between the frequency of variants and its effect. Now, just imagine that each of those 3 million variants per individual has arisen at some time in the past as a mutation. So it has arisen on a single genome and has been passed on from generation to generation. So until this variant spreads through the population to a considerable frequency, it has to be passed on for many, many, many generations. That means that the effect of the variant that has been passed on for hundreds of thousands of generations must be relatively small. A strong effect variant, for example, one that causes a severe disease, cannot be passed on for many, many generations because it would be evolutionary detrimental. So in general, common variants are usually very ancient, but they have a small effect. Large effect variants are usually relatively recent. They're either de novo, they just arise as a new mutation, or they're relatively recent in the evolutionary history. So that's interesting for the reason that this also can be applied to genetic diseases. And we see a general pattern of the architecture of complex diseases, and I will show you in a minute how this applies also to Parkinson's disease. We have generally rare alleles, very rare alleles, that have a high effect, and those are the ones that cause Mendelian forms of the disease. So if you have the mutation, if you have the variant, then you will develop the disease. There's an intermediate class, low variant frequencies with an intermediate effect. Those are risk factors, and we will see what that means in the case of Parkinson's disease. And then there are relatively common variants that occur in 10, 15, 20, or even more % of the individuals. They can be risk variants for a disease, but they have a relatively low effect. Their effect strength is on the order of 1.1 or 1.2, so they only increase the probability to develop a disease by a few %. The rare alleles increase the probability of a disease by a lot. That's a very famous graph here that's usually used. It's a Manolio graph that is attributable to many diseases, for example, also to Parkinson's disease. And here we will get to know some of those variants during the talk. Some of the rare variants are called alpha-synuclein or Parkin or PINK1 or LARC2. Those are genes that harbor rare, high effect strength variants. There are some of those intermediate classes, and GBA, the glucocerebrosidase gene, is one of the major ones, and the one specific mutation of the LARC2 gene, the G2019S. And then there's a large number of risk variants of low effect present. We have detected about 90 of those risk variants. So that's a general architecture of Parkinson's disease, and I will come back to this pattern during the talk several times. So let's start with the actual Parkinson's disease problem. So we know that those Parkinson's patients have a specific clinical phenotype. So they have a small stepped gait, they have a stooped posture, they have tremor. And we know that this is caused by the degeneration of the substantia nigra dopaminergic neurons, the lack of dopamine in the projecting areas, and they have also specific changes in their histopathological picture, namely the Lewy bodies and the degeneration of the appropriate dopaminergic neurons. We can treat those patients with drugs with dopamine replacement or with deep brain stimulation. And that's a patient who has been treated now, he's turned on his stimulator, and you can see that his gait improves. He walks briskly with normal steps, not completely normal. And you can see that is slightly impaired, but also his postural stability has improved. So we can treat the symptoms of those patients relatively well. The genetic history started about 1997, really, the modern genetic history of Parkinson's disease. Until then, we thought that Parkinson's disease was not a genetic disorder. In 1997, Bob Nussbaum here discovered in a family that was identified by Larry Golby in the south of Italy, where they had their origin in the so-called Contursi kindred, a mutation in a gene for alpha-synuclein. Very little known gene at the time, alpha-synuclein, but turned out that a point mutation in this gene is the cause for Parkinson's disease in this large family with an autosomal dominant inheritance pattern. Now, in this family, Parkinson's disease was inherited as a dominant disease. So you can see here the little black dots go from generation to generation. About 50% of the individuals, of the offspring, actually develop the disease. But they developed the disease relatively early. So the average age of onset was 44 years. They had a rapid progression and frequent dementia. So at that point, after the discovery of the gene, a lot of people thought, well, is this really Parkinson's disease? Is this the same as our normal Parkinson's disease that we see in the clinic? And a lot of people doubted that until just shortly afterwards, these guys, Maria Grazia Spilantini and Michel Goedert, discovered that the Lewy bodies, the intracellular inclusions that are typical also in the sporadic forms of Parkinson's disease, they are made up out of this protein, out of the alpha-synuclein protein. So they used an antibody to alpha-synuclein and could show that they stained the alpha-synuclein in those in the brains of Parkinson's disease patients, not only those with mutations, but also those without mutations in this gene. So that puts alpha-synuclein at a very central spot in the Parkinson's story. A little later, Andy Singleton, at that time at the NIH, discovered in another family that looked very much like this original alpha-synuclein family, dominant inheritance, early onset, that not only point mutations, but also a multiplication, in this case, a triplication of the gene, caused autosomal dominant Parkinson's disease. You can see here that's the so-called fluorescence in situ hybridization. So it's a method to visualize the genes on the chromosomes. And you can see that on one chromosome, there is one gene, and on the other chromosome, there are 3 genes. So it's a total of a duplication of the gene dosage. Usually you have 2 genes. If you have 3 copies on one gene, you have a total of 4 copies. So it's a strong overexpression of the alpha-synuclein gene, and that also causes autosomal dominant Parkinson's disease. And a little later, it was discovered that also gene duplications also cause Parkinson's disease. So for this gene, we now have the situation, which is very clear. Both mutations that increase the propensity of the protein to aggregate and overexpression causes Parkinson's disease by aggregation of the alpha-synuclein in so-called Lewy bodies. Now, the only problem is that only 1% or even less of Parkinson's disease patients carry point mutations or multiplications in this gene. But all Parkinson's patients have these alpha-synuclein aggregations. So for the alpha-synuclein, the situation is actually very similar to Alzheimer's disease, that alpha-synuclein point mutations and alpha-synuclein overexpression leads to protein aggregation and to Parkinson's disease. Very similar in Alzheimer's disease, where rare mutations in the APP gene and the overexpression of the APP gene, which is found in trisomy 21, for example, causes Alzheimer's changes. So for a very small segment of patients, those with point mutations, those with overexpression due to duplication, the story seems to be solved. The problem is that's only a very, very small percentage of patients. And so I told you that was 1997, 1998. Of course, research went on. And now we know that a large number of mutations in very different genes with affecting very different molecular pathways can result in the same disease and in the same effect. And you see a slightly incomplete list here. There are some more genes, but they're getting rarer and rarer. And you can see that alpha-synuclein is on there. The red ones are the ones that are autosomal dominant. We call that gain of function. The green ones are autosomal recessive. We call that loss of function. And they affect different cellular pathways. The alpha-synuclein is apparently related to protein aggregation. The green ones, as you will see later, are related to the mitochondria, to mitochondrial quality control. LRRK2, also an important gene, is probably related to vesicle transport and vesicle sorting. And the GBA is a gene that is important in lysosomal function. So we will discuss in a little more detail those three genes, Parkin, LRRK2, and GBA, as examples of those pathways that lead to Parkinson's disease, even if the alpha-synuclein gene is not affected itself. The alpha-synuclein gene, however, comes downstream and is probably related to Parkinson's disease in the vast majority of cases. All right, so I show you another example, case report. This is a patient. My speaker off, okay. So this is a patient who you can see looks quite different from the one that I showed you in the first instance. He's very young. You can see his face or part of his face in a minute. Here you cannot really recognize him, but you can see that he's very young. He's 38 at the time of the video. Onset of disease was 31. He has a typical levodopa responsive Parkinson's disease, quite akinetic here. I can maybe run that again. You can see the loss of arm swing. Gait is relatively fluent. He's on levodopa therapy, and in his good times, he always also developed dyskinesias, but no cognitive impairment. He has a MoCA score of 29. No family history. So in a talk with the audience present, I usually ask now, do you have an idea what could be the related gene? Now we can do that, but you can probably think about it, and maybe some of you will know. This is a patient with a compound heterozygous mutation in the Parkin gene. So that is one of the recessive early onset or juvenile Parkinson's disease genes. And you can see that this autosomal recessive inheritance means that both parents carry the mutation, but only individuals that inherit both mutations will develop the disease. In the meantime, we have found a large number of recessive Parkinson's disease genes that cause slightly different forms of the disease. The green ones here are the ones that cause relatively pure Parkinsonism. Early onset Parkinsonism, no or little cognitive impairment, early dyskinesias, That's a typical clinical picture of a recessive Parkinson's disease. Onset mostly 30 or even younger. And then there's another class of recessive genes that cause complicated Parkinsonian syndromes. So they usually have different other symptoms like spasticity, like dystonia, like cognitive impairment, and we're not going to talk about them in more detail. The ones that are related to the pure Parkinson's disease, we have at least some conception of what these genes do that could cause the detrimental effect on the dopaminergic neurons. We know that at least two of them, parkin and pink1, have an important role in the maintenance of mitochondrial function. You know that mitochondria are the important cellular organelles that provide energy to the cell, and they're relatively dangerous. They're little time bombs in the cell. They can be damaged, and when they're damaged, they leak toxic oxygen radicals. So the cell needs a machinery that removes damaged mitochondria relatively rapidly. And parkin and pink1, the two Parkinson's disease genes, are involved in this process that is called mitophagy. It's the removal of damaged mitochondria from the cell. And if those genes are lacking, then we think that the sequestration of the damaged mitochondria is not guaranteed in a timely fashion. And that leads to damaging of the dopaminergic neurons, because dopaminergic neurons have a high level of energy requirement. So they have a lot of mitochondria, they have a lot of energy consumption. And that's why when the mitochondria are damaged and they cannot be removed, the consequences are detrimental to the cell. And that, in these cases, leads to the death of the dopaminergic neurons. That's at least the current hypothesis. So what we could do is, of course, and that's background of much of this research, identify those individuals and try to treat them by stabilizing their mitochondria specifically. And with Christine Klein in Lübeck, we're actually involved in such a study that we use parkin homozygous or compound heterozygous mutation carriers, heterozygous mutation carriers, and those who have a genomic pattern that resembles this mitochondrial form of Parkinson's disease, and treat them with coenzyme Q10, which is a drug that supports mitochondrial function. And what we will be studying is not only the effect on the function of the neurologic symptoms, on the development of the neurologic symptoms, but also on the energy production in the brain using specific MRI sequences. So this is a form of targeted treatment, targeted to the enzyme, to the mitochondrial deficiency of those juvenile Parkinson patients. The study is not completed yet, and we'll see if that has any effect. That's another case of a monogenic form of Parkinson's disease, which is, again, quite different. That's a female patient. At the time of the video, she was 64. Onset was 55, so it's slightly on the younger side for Parkinson's patients. At that point, 9 years into the disease, she's treated only with the dopamine agonist, pramipexole. She has no dyskinesias. You can see here that she's doing all those exercises very fluently. She has, on treatment with the dopaminergic agonist, very few symptoms. She has no cognitive impairment, no family history of the disease. This is also a genetic form of Parkinson's disease, even though she doesn't have a family history. And it turns out that this patient has a LAG2 G2019S mutation. Now, what's characteristic of the LAG2 mutations? One is characteristic one characteristic is that the effect of the LRRK2 mutation is what's called pleomorphic. So it causes different phenotypes, at least pathologically. This is one of the first large families where this gene was discovered. And pathologic examination shows that those affected individuals have, in fact, different pathologies. So one had a typical brainstem Lewy body form of Parkinson's disease. One had cortical Lewy bodies, like in Lewy body dementia. One had just extracellular pigment, but no Lewy bodies. And one even had tau pathology. So variable pathology, but all have clinical Parkinsonism, dopa responsive, and dopaminergic degeneration. So not all of them, it's the same mutation, but not all the same pathology. The other interesting thing with the LRRK2 gene is that at least one mutation, the G29S mutation, the one that also was found in our patient that I just showed you, is relatively frequent in certain populations. So it's not as common here in Northern Europe. It's only 2%, 1 to 2% of Parkinson patients have this mutation, the same in the US. In Southern Europe, it's a little more frequent, up to 7%. But in Israel, in the Ashkenazi Jewish population or in the North African population, it is very common, and it's up to 30% of Parkinson's patients have this G2019S mutation. Not all of them have a family history. So this is one of those genes that has a moderate effect and a reduced penetrance. And you can see the penetrance, estimated penetrance varies in different studies, but the average is probably around 30 to 40% penetrance at the age of 80. So not everybody with this G2019S mutation develops Parkinson's disease, only a fraction of them, and the others stay healthy throughout their life, but they pass on the mutation to the next generation. So that's a peculiarity of this G2019S mutation. What does the LRRK2 gene do? It's a very large gene. It was discovered in 2004, but the function was unknown for a very long time. And only in 2018, it was discovered that the LRRK2 gene is a kinase that has other intracellular proteins as substrates, the so-called RAP proteins, which are also signaling proteins in the cell. So the LRRK2 gene is involved in a signaling cascade by functioning as a kinase. So that's the G2019S mutation here, but there are other mutations in the LRRK2 genes that cause an increase of the kinase activity that phosphorylates these RAP proteins. So it's an overactivity. That's why it's called a gain of function. It's a gain of the kinase function. And this gain of kinase function, again, affects a lot of cellular processes like mitochondrial function, microtubule, microvesicle processing in the cell, autophagy, which is a protein removal function in the cell. So the effect of the LRRK2 gene are very variable, but the function is simply an increase in its kinase capacity. So LRRK2 gene hyperphosphorylates other proteins, and that leads to the dopaminergic degeneration. And therefore, again, a targeted treatment can be thought of, a kinase inhibition as a treatment in patients with LRRK2 mutations, but possibly also in other Parkinson's disease patients, because there is evidence that the kinase activity can be overactive even in the absence of these pathogenic Parkinson's mutations in PD patients. So this may be a unifying effect or phenomenon, the LRRK2 kinase overactivity, and therefore a lot of companies are presently developing LRRK2 inhibitors, probably not without side effect, because the LRRK2 function, the kinase function, is important. But that's, of course, in many diseases, this has to be considered, and a balance of overactivity and underactivity has to be met. So that's another potential targeted mutation-specific treatment that can benefit potentially also other Parkinson patients. Okay, we come to the third monogenic form of Parkinson's disease. Here's still another patient. It's again different from the others. That's the oldest one that we have in our collection. He has an age of 70 at the time the video was taken. He had an age of onset of 60, so typical Parkinson's disease, but he has relatively rapid progression. At that point, he's on medication, but you can see that his very small step, he has severe resting tremor. He also had cognitive impairment, also orthostatic dysfunction. So when he's standing for a longer time, his blood pressure drops. And his brother had Parkinson's disease. So he's a patient with a complicated, relatively rapidly progressing Parkinson's disease with cognitive impairment and orthostatic dysfunction. Think for a moment, what gene could it be? We cannot discuss that now in the audience, but it's a GBA L44P mutation. It's one of the severe glucocerebrosidase GBA mutations. Now, GBA is a gene that actually causes Gaucher's disease if it's homozygous, if both genes are mutated, but it increases the risk for Parkinson's disease by a factor of 3 for the mild mutations, up to 20, 15 or 20 for the severe mutations. So that's one of those risk factors that have a strong effect. And it's actually relatively common in the German population, and about 7 to 8% of Parkinson's disease, or overall in the European population, 7 to 8% in our hands of Parkinson's patients have variants in this GBA protein. So are heterozygous mutation carriers for this Gaucher disease gene. And as you could just see in the video, these GBA mutation carriers have a more rapid progression. They have earlier dementia. They have more autonomic symptoms than the average Parkinson patient, and also compared to the LRRK2 Parkinson patients, which tend to have a somewhat milder form of Parkinson's disease. What's the function of the encoded gene, of the glucocerebrosidase, which is encoded by the GBA gene? It's a protein that is involved in lysosomal function. The lysosome is an organelle here that is involved in the degradation of protein, and is believed that abnormally folded synuclein is degraded by the lysosome. And if there is a mutation, that's a normal case. If there's a mutation in the disease case, there's no function, or there's an impaired function of the lysosome, and therefore the alpha-synuclein aggregates, and the cells that produce a lot of alpha-synuclein oligomers and aggregating fragments become damaged, and Parkinson's disease develops. So again, a specific process that is linked to the development of Parkinson's disease caused by the mutation of this glucocerebrosidase gene. Again, there are options of mutation-directed treatments. For example, one could, this is actually the pathway. So there's the block due to the glucocerebrosidase mutation. One could try to restore the enzyme function. That's not easy, because enzyme replacement usually doesn't cross the blood-brain barrier. So the trial that was actually already done was to start at another point in this pathway by inhibiting the substrate formation, so the synthesis of glucosylceramide by a glucosylceramide synthase inhibitor, venglustat. And that trial was partly effective. It was done, it was effective in reducing the substrate of the glucocerebrosidase, but in this trial that was done by Sanofi, it failed to reach the primary endpoint, namely to clinically improve over placebo the clinical condition of the patients. So while this concept is still, and I think still should be considered interesting and promising, so far it failed, possibly because the effect of substrate reduction was not strong enough in the CNS. This is measured in the serum. There, the substrate reduction was very strong, but in the CSF, it may not have been sufficient. So I've showed you 3 different types of monogenic disease forms of Parkinson's disease that can be addressed by, or that have been addressed by trials of targeted treatments, the mitochondrial form with Parkin mutations, the LRRK2 form with a LRRK2 kinase inhibitor, and this GBA-related form by substrate reduction. We have to remember, though, and keep in mind that all these forms of monogenic Parkinson's disease that we just discussed are only, show this here, the tip of the iceberg. They are the ones where we find mutations, and they're probably less than 10% of all patients that have one of those mutations that we've discussed so far. So what about the 90% of sporadic cases? And although this is a talk on the genetic forms of Parkinson's disease, which I've basically done now, I will spend a few final minutes on showing that the sporadic, or what we call idiopathic Parkinson's disease, also has important components that are related to the genes that we just discussed. Now, we cannot, of course, identify the genetic component in those sporadic cases by studying families, because there is no family history of the disease in the sporadic cases. But we can study it by genome-wide association studies, by comparing cohorts of patients with Parkinson's disease and controls. And these association studies basically look for genetic variants, comparing those with mutation to those who age healthy and without Parkinson's disease, grow to a high age without Parkinson's disease. And the variants that are more frequent in the population with Parkinson's disease have some probability of being associated with the disease and contribute to the causation of the disease. And that is actually, that can actually be shown by those genome-wide association studies. That's one of the first that was published. And you can see that you need a lot of cases, 1,000 cases, 3,000 controls in the exploratory phase. Then you do a reproduction, a confirmation phase, and so on and so forth. And in this first study that was published in 2009, there were only 2 risk factors that were identified. And that's called a Manhattan plot. That's a depiction of the genomic, the significance of the association of a variant along the entire genome from chromosome 1 to the X chromosome for being a risk factor for the disease. And you can see that the alpha-synuclein here is a risk factor, and the tau gene is a risk factor. That was only the first study. In more recent studies with many, many more patients, over 37,000 patients, 1.5 million controls, so a lot of patients, there are a total of 80 loci that have been identified. And interestingly, you can see the GBA gene, the alpha-synuclein gene, the LRRK2 gene, and others show up here again. So as I showed you initially, the link between the rare and the common variants also holds true for Parkinson's disease. And we can see that the same genes are involved with rare variants in monogenic forms of Parkinson's disease, and with the common variants that are investigated in GWAS studies, for the common sporadic form. And in order to just have, I have 3 or 4 more slides, which I will skip now because they go more in a little bit of detail of the effect of those common variants. But to give us a little more time for the discussion, I would like to end up with a conclusion. Conclusion being that Parkinson's disease, as you can see, has a complex genetic architecture with common and rare variants influencing different aspects of the phenotype, like age of onset, development of dementia, and so on. They are very different, the different genetic forms of Parkinson's disease. Different genetic molecular pathways are involved in the development of the disease, but also risk variant patterns in the sporadic disease are involved with in the sporadic forms, that what we can find from the genome-wide association studies. And the therapeutic potential is probably highest in those with the intermediate effect strength variants, so in the GBA gene, in the LRRK2 G29S genes, where we can find enough patients who have those variants, but who have a defined genetic variant that can actually be addressed by genetically directed treatments. And with that, I would like to conclude and be happy to answer questions. There are just some acknowledgements from my lab and other collaborators who we have been working with for a long time. So thank you for your attention.